Hello students, in today's class we are going to learn about apoptosis, also known as programmed cell death. So apoptosis is a pathway of cell death that is induced by a regulated suicide program. The dying cells activate intrinsic enzymes that degrade cells' own nuclear DNA and nuclear and cytoplasmic proteins. The apoptotic cells breaks into fragments known as apoptotic bodies that bleb out from the cell, and these blebbing bodies are eaten up by the uh, macrophages, preventing the inflammatory response. On the other hand, necrosis happens, which involves activation of an inflammatory response because of the leakage of the cytoplasm. So basically, there are two different pathways of apoptosis, extrinsic and intrinsic pathways. So these are the some of the features of apoptosis. So this is an ATP dependent program cell death. Two pathways, extrinsic and intrinsic pathways are involved. Both pathways activate a caspases that are basically cytosolic proteases. So here we have cellular breakdown, including cell shrinkage, chromatin condensation, membrane bleeding, and formation of apoptotic bodies, which are then phagocytosed. The cell membrane remains intact without significant inflammation, which is not the case with necrosis. And there is also certain changes in the cell membrane composition that are being identified. For example, one of the lipid phosphatidyl serine is present in the inner leaflet of the cell membrane in normal cells, while in the apoptotic cells, it switches or it flips to the outer leaflet. So, which is a distinguishing, identifying characteristic of a apoptotic cell. Then a DNA laddering like feature is also observed. That is because of fragmentation of the DNA into smaller sequences, about 180 base pairs in length. Now coming to the actual pathway of apoptosis, there are basically two identified pathways for apoptosis. The first pathway is the extrinsic pathway, which involves specific DNA ligand interaction. For example, we have the TNF receptor and the Fas ligand interaction that can mediate the extrinsic pathway. This triggers and initiator caspase. So the initiator caspase is cleaved, and that leads to activation of the initiator caspase. So this cleaved caspase will further bring about cleavage and activation of the actual executor caspase, that is caspase 3, which gets activated on cleavage. So the caspase 3 will then bring about the downstream apoptotic response, such as the formation or blebbing of the cells, degradation of the DNA, and then these bleb cells are being engulfed by the macrophages. Secondly, we have the intrinsic pathway. So this pathway basically involves the role of mitochondria. So this pathway is triggered by sensing some kind of damage, such as the DNA damage. This damage is recognized by specific sensors, such as the BCL2 family sensors that can recognize the DNA damage. Then we have the BCL2 family effectors, such as the BAX and the BAD proteins that can poke holes in the mitochondria and will lead to release of C cytochrome into the cytosol. Then specific enzymes such as APAF1 interacts with the C cytochrome, and that will lead eventually into again activation of caspase 3 by cleavage, which will further bring about the uh, downstream apoptotic response similar to uh, that induced by the extrinsic pathway. So here we also have the BCL2 and the BCLXL proteins that promote cell cycle proliferation uh, by inhibiting the activity of the BCL2 family effectors. So we have these same points written in the slides for your reference for the extrinsic pathway as well as for the intrinsic pathway. So that's it about apoptosis. So kindly like, share, and subscribe if you like the contents, and thank you.